Welcome, Dr. Palushaj. She's a senior neurology resident at Stanford University in the fellowship program for movement disorders. Today, she will be speaking about microbiome, the microbes that live in our guts, and Parkinson's disease. Studies show that the microbiome in Parkinson's patients is different from the microbiome in healthy individuals. What is not clear, however, is how these differences impact one's risk for developing Parkinson's disease or the course of the disease. Dr. Palushaj is the principal investigator of a study to better understand the mechanism by which the microbiome in Parkinson's patients may be contributing to the disease. In the future, these findings could be used to develop disease treatments that target the microbiome. They are recruiting individuals who have Parkinson's disease and healthy individuals from the same house. Participation entails answering an online questionnaire. You will receive a $40 gift card for your involvement. Clinical trials so often require visits to distant institutions, but this is one that they are bringing to us. Stanford will send a phlebotomist to take blood samples at our March 11th general meeting at the church, 1717 Yalupra Avenue, from 11 a.m. to 1 p.m. immediately prior to our next meeting. Those interested in participating can fill out the short form that will be offered to you. After filling it out, the Stanford team will reach out directly to get the consenting process started. So to just reiterate, any of you who want to be part of this process can be involved, and we'll be getting blood samples at our next meeting. It's a very exciting clinical test. Thank you.
Thank you so much, Tess. I appreciate that. Well, thank you so much for having me here, you guys. I'm extremely excited to be here and to share our work with you and to talk about the microbiome and Parkinson's disease. So thank you for this opportunity. I actually went to medical school late, so I had some years out of my undergraduate and medical school, and I worked at 23andMe back in the day when they were a very young company. And I actually was hired at that time to help manage the Parkinson's disease cohort. And so this was almost 15 years ago, I think, at this time, but I used to travel and talk to Parkinson's disease support groups at that time, kind of freshly out of college. So it's really a fun honor to be continuing to do that from a different perspective these days. So again, my name is Bianca. I'm a neurology resident at Stanford, and I'm staying to do a fellowship in movement disorders for the next 2 years at Stanford. And of course,
make the captions bigger.
My research interests are in the microbiome and Parkinson's disease. So without further ado, let's get started. First of all, why should we study the gut in Parkinson's disease? I don't need to tell you guys that about 60 to 80% of patients experience GI symptoms years or even a decade or more before the onset of stiffness and tremor and the more classic motor symptoms. And so, of course, you guys are familiar with constipation, may start up to 10 years before those motor symptoms. Smell problems. Some patients might notice difficulty with sleep, REM behavior disorder, mood changes, etc. And so this suggests to us that Parkinson's disease may actually begin in the gut before traveling from the gut to the central nervous system or the brain. So what do we know about the gut and Parkinson's disease? First, I just want to define alpha-synuclein. This is the protein that is really the hallmark of Parkinson's disease. And we get aggregates of alpha-synuclein, or kind of clumping of this protein, that becomes toxic to the neurons and can then lead to neurodegeneration. And so what we know today is that alpha-synuclein, this abnormal protein, can be seen in the nervous system of the gut that's called the enteric nervous system in the very early stages of disease. So again, in the stages long before motor symptoms develop. There have been studies that have shown that when animals have alpha-synuclein injected into the intestinal wall, you can then see that alpha-synuclein travel from the intestinal wall, from the intestinal wall through the vagus nerve up into the central nervous system and the brain. And then, of course, there was a famous Danish study from 1977 to 1995, and that showed that individuals who have a vagotomy, where the vagus nerve is cut, have a much lower risk for developing Parkinson's disease, suggesting that the vagus nerve may be really critical for the pathogenesis or the development of Parkinson's disease. So that's the gut. And what about the microbiome? First, let's just define what the microbiome is so we're all on the same page. The microbiome is a collection of microorganisms that live inside of us and on us. It consists of bacteria, viruses, fungi, archaea. It's thought that bacterial cells actually outnumber human cells by 10 to 1. And I have a little asterisk here because this is kind of a hotly debated ratio. With newer technologies, some people are actually arguing that it's more than this. And then there's some debate as to, how we define a human cell, whether it's through nucleated cells or not, which would not include red blood cells. But generally speaking, we have about 10 times more bacterial cells than we do our own human cells. Furthermore, bacterial genes outnumber human genes by a factor of about 1,000 to 1. And so classically, traditional microbiology, the field of microbiology, focused on studying one bacterial species. And in order to study each individual bacterial species, we had to culture them in a petri dish. But then in the mid-2000s, there was advances in DNA sequencing technologies, and that really led to this new field of research that's now called metagenomics. Metagenomics allows for a more comprehensive examination of not just individual species, but of whole microbial communities. And importantly, we're able to do this without culturing these microbes. This is really important for microbiome research because the vast majority of microbes that live inside of our guts are not able to be cultured in a petri dish. So this really opened up a lot of opportunity for studying the microbiome. The Human Microbiome Project was led by the National Institutes of Health, and that went from 2008 to 2013. It cost the US $115 million, and it included 242 healthy adults and took samples from 5 different body sites. And the goals of this project were just to characterize the microbiome and to begin to analyze its role in human health and disease. So from the Human Microbiome Project, and then, since then, we've learned a lot about what the microbiome functions. And so we know now that the microbiome produces certain vitamins that we don't have the ability to make on our own. We don't have the genes necessary to produce some of these vitamins. And so we rely on the bugs inside our gut to help us make those vitamins. The microbiome helps to break down our food and help us extract critical nutrients from our food. The microbiome is really important in teaching our immune system how to recognize, who's dangerous and who's good. So there's a big association between changes in the microbiome and changes in immune system functioning. And likewise, it produces anti-inflammatory compounds as well. We know that the microbiome is really important for producing healthy gut motility. And of course, the other side of that coin is that there are some characteristic changes in the microbiome that are associated with constipation or delayed gut motility. Finally, the microbiome engages in what we call bug-on-bug warfare, where we have very important commensal or good bacteria that help fight off disease-causing pathogens. The photo on the right represents one of my favorite facts from the animal kingdom, and this is that when baby elephants are born, they don't have the necessary microorganisms in their guts in order to digest plant materials. And so the only way for them to get these gut bacteria so that they can then digest the plants that they eat is to actually eat the feces from their mother or from other adult elephants in their herd. And so this is something that we see baby elephants do, is eating the feces when they transition from a milk to a plant-based diet. So how does the brain and the gut communicate? And what is that interaction? There's at this point thought to be several mechanisms by which they communicate. So microbes that live inside of our gut produce metabolites. And so byproducts of their metabolism can be metabolites that either can directly act on the brain. And one example of this is something called short chain fatty acids. Short chain fatty acids are a byproduct of, again, microorganism. When they ferment carbohydrates, they make short chain fatty acids. Short chain fatty acids are thought to be really critical for the importance of the health of our colon and the cells that live inside of our colon. They also impact microglia cells. Microglia cells are they're immune cells that live just in the brain. They're unique to the brain, and they're actually very much implicated in the pathogenesis of Parkinson's disease. And so again, to summarize that whole concept is that the bugs in our gut, through their metabolism, produce these products that can either directly have effect on the brain, or the bugs can kind of produce this chain of events that produce molecules like neurotransmitters, hormones, vitamins that then, act both directly on the brain and in other physiologic ways inside of our body. Another mechanism that the gut and the brain interact is through gut permeability. So leaky gut, you may have heard that term, or increased gut permeability, has been associated with a lot of systemic diseases at this point. And we think that when there's inflammation inside of the gut, in part because of an altered microbiome, that inflammation allows for increased gut permeability. And with that increased permeability, microbes, as well as the metabolites that they produce, can leak from the inside of the gut lumen into our bloodstream and then cause more systemic effects. Finally, we've talked about the vagus nerve. And so the vagus nerve is really quite literally a direct communication from the enteric nervous system, which has more neurons. We actually have more neurons in our gut than we do in our brain. And the vagus nerve connects that enteric nervous system from the gut to our brain, allowing for a direct communication. So what we know about the microbiome and disease. So I talked about the Human Microbiome Project. That really was in healthy individuals to just begin to understand what the function of the microbiome is, even in healthy individuals. Since then, there's an ever-growing number of studies that have shown that changes in the composition of our microbiome is associated with any number of diseases. So, there was probably a span of about 10 years where the microbiome was, we were just starting to study it, and study after study was coming out saying there's changes in the composition of the microbiome in multiple sclerosis compared to people without multiple sclerosis, Parkinson's disease compared to people without Parkinson's disease, Alzheimer's disease, et cetera, et cetera. But then furthermore, we've also started to understand that these perturbations or changes in the gut microbiome is not just, seen when you compare people with a disease to people without a disease, but we think that they can also, that they, as in the changes in the microbiome, can also change one's risk for developing a disease. They're implicated in the disease activity and how the disease progresses, as well as the phenotype. So for Parkinson's, that might be, whether you have more of a motor predominant Parkinson's disease or not, whether GI symptoms are very predominant for you or not, whether you have cognitive symptoms, etc. So I just want to summarize kind of where we're at with the microbiome and Parkinson's disease. What do we know today about the microbiome and Parkinson's disease? As I've alluded to many times, it's very well established at this point that the composition of the microbiome in Parkinson's patients is different than non-Parkinson's patients. So time and time again, we've shown that when you compare the microbiome in people with Parkinson's disease to people without Parkinson's disease, there's some really substantial differences in which bugs are there. Some bugs are enriched, some bugs are depleted, and I'm just going to check the chat because, we were having trouble, Stuart, with showing just the slides, so we decided to just get going. I'm sorry, I got a question about the slides. And so we know that the composition is different. In the last couple of years, what we as in the scientific community has been trying to do is show beyond the composition, what is the significance of that? So not just who is there, but what are they doing and how is this causing differences in function that's impacting human health. So I talked about short chain fatty acids. Again, this is about Parkinson's patients have a bacteria that short chain fatty acids. This has a ton of implications. Again, short chain fatty acids are important for the health of our colon and the colon, but importantly, may influence the microglia cells, which again are those immune cells that live inside the brain and may make them more inflammatory. It's also been shown that the microbiome in Parkinson's patients causes an increased production of inflammatory metabolites. So there's more kind of bad players, certain types of bugs that tend to be more pro-inflammatory. And then there's also been studies that show that E. coli is a type of bug that almost all of us harbor, but there are different strains of E. coli, and some strains of E. coli produce a protein that's called curly. And it was shown that this curly protein that's made by the E. coli bug can actually cause the aggregation or clumping of that alpha-synuclein protein, leading to insoluble tangles of that protein that becomes toxic to the neurons. So, what can we do about it? How can we fix it? What's next? This slide is really just to remind ourselves that there are many, many, many contributors to the microbiome. It's not as simple as just changing diet or taking a probiotic or a prebiotic. I wish it was. So again, just keeping in mind that we can't be so naive to think that changing one or two variables is really going to make lasting change. And then also just keeping in mind that when we compare, again, groups like Parkinson's patients and non-Parkinson's patients, and we look at their microbiome, we have to keep in mind that there's many other things that might be influencing those changes in the microbiome. So I was asked to talk about some diet recommendations as well as some supplement recommendations. And of course, these are common questions we get in clinic all the time. There are really two main pillars of diet that I want to talk about here. And one is, what should I be eating? Is there a diet that's best for me? And two, some dietary practices to address the fact that most, if not all, patients with Parkinson's disease have what we call gastroparesis, or delayed gastric emptying, slow gastric motility. And so there are some things that we can recommend, both how we eat and kind of some lifestyle changes that can help support that gastroparesis. I will caveat these diet slides with saying that this is not evidence-based. There's kind of a paucity of randomized controlled trials studying diet in general, as well as diet in Parkinson's disease. So this is my own personal philosophy and recommendation based off anecdotal evidence and talking to colleagues both in the GI clinic as well as neurology clinics. For those of you who aren't familiar with Dr. Andrew Weil, he is a doctor so he studied biology at Harvard. He studied botany, actually, and then went on to get his medical degree at Harvard. He is really a proponent of preventive medicine and natural medicine, and he founded the Integrative Medicine Center at the University of Arizona. And this on the right is a picture of Dr. Weil's anti-inflammatory food pyramid. On the left, this is contrasted with the standard USDA food pyramid. And what you'll notice with Dr. Weil's food pyramid is really at the base of it is fruits, vegetables, grains, but more non-processed whole grains, as well as healthy fats and then proteins, with more of a focus on fish and proteins that are high in those healthy omega-3s than red meat, though not to say that you can't eat red meat or poultry. Just to look at, so this can be found online if you guys want to, look at this a little more closely. Again, just Dr. Andrew Weil's anti-inflammatory food pyramid. And just to go through some of the tenets of that, starting at the base. So for fruits and vegetables, the thing that you want to think about with fruits is trying to eat fruits that are lower in glycemic load. This tends to be berries as well as stone fruits. It's usually tropical fruits that have the highest glycemic index. For vegetables, the main principle here is that you want to try to consume diverse amount of vegetables. So what we were told when we were kids, eat the rainbow, eat a wide variety of colors. That is true. So diversity, a lot of colors, lightly cooked, dark leafy greens, cruciferous vegetables. Often forgotten are sea vegetables, which have important sources of iodine and other important micronutrients. And for individuals with gastroparesis, you want to avoid raw vegetables. And we'll talk more about why that is. But again, lightly cooked is the best method for that. Proteins, really, a variety of proteins, cheese, yogurt, eggs are also high in omega-3s. Poultry, we recommend skinless poultry. And then red meat is fine and great, but I recommend grass-fed and grass-finished. Salmon, the biggest bang for your buck in terms of nutritional status with salmon is sockeye salmon, and then herring, sardines, and cod. A lot of these are, lower mercury and high omega-3 fishes. For fats, the fats that we like are those that are rich in either monosaturated or omega-3 fat. So extra virgin olive oil, nuts, avocados, seeds, flax seeds, hemp seeds, etc. So, you can get in the weeds. Diet can be complicated. There's a lot of things to consider. I still love this quote by Michael Pollan, and that is, eat food, not too much, and mostly plants. And it has some good basic principles here. Eat food, try to avoid processed foods, packaged foods, not too much generally, especially with gastroparesis, eat to about 75% full, and do include plants. And we'll talk about for individuals with slow gastric motility, how to do that for yourself to minimize kind of bloating and abdominal pain. Another thing to keep in mind with diet is trying to minimize pesticides, herbicides, and eating organic when possible. And I recognize that those of us who are able to eat organic, it's has financial implications, and we're very fortunate to be able to eat organic. And we also live in California, which is another fortune for us. But you can find this list of what's called the Dirty Dozen and the Clean 15 on the website for the Environmental Working Group, or EWG. But the principle here is that these 12 that you see in the red are tend to have the highest residue of pesticides on them. And so when you can, it's really important to try to eat those in the organic form. On the right, you have in blue the Clean 15, and those are the ones that there really isn't too much of a difference in chemical exposure and chemical load between the organic and the non-organic. So we think that it's generally okay to eat the non-organic. I'm always happy to see that asparagus is on there, because if you look at the price difference between organic and non-organic asparagus, it's quite substantial. I think this is really interesting, especially given that, we don't know a ton about risk factors for Parkinson's disease and what makes someone higher risk for developing the disease. But the one thing that we do know is that there's an association between exposure to chemicals, pesticides, and herbicides and increased risk for Parkinson's. So this is something that I think is really important for Parkinson's patients. Another diet principle is to remember to eat fermented foods. And this can include yogurt, kefir, fermented cottage cheese, though I feel like I don't see that very often at grocery stores, kombucha, tea, other fermented vegetables like kimchi. There was a study out of Stanford University, Justin Sonnenberg's lab just last year, looking at the impact of fermented foods on the microbiome and also on systemic inflammatory markers. And this study showed that there was an increase in diversity of gut microbes in the microbiome from eating 2 servings of fermented foods per day. And there was also a decrease in inflammatory markers. So pretty exciting and substantial that just by eating 2 servings of fermented foods per day, they could actually detect a decrease in inflammatory markers in the blood of patients. And Christopher Gardner is a professor, and he also is the director of nutrition studies at Stanford Prevention Research Center. And he said nicely that microbiota targeted diets can change immune status, providing a promising avenue for decreasing inflammation. And so here he's referring to a microbiota targeted diet being fermented foods can change one's immune status. So then to move on to some of the dietary practices and kind of lifestyle approaches to support gastroparesis. You are probably aware that it's best to eat smaller and more frequent meals. The patients with gastroparesis will have a harder time digesting a larger meal. Keep in mind that digestion begins with chewing, and so it's very important to chew well, something that I all too often forget. You want to minimize fat, and minimize, not eliminate, but minimize fat, because fat can delay the emptying of the stomach. That's also, the benefit of why we feel more satiated and full for longer when we eat healthy fats. But just keep in mind that a higher fat meal will sit in your stomach longer. And again, you want to avoid raw foods and foods that can't be chewed well. And that's really because, this is true for anyone, even those without delayed gastric emptying, but especially for people with delayed gastric emptying, raw foods can really be unsettling and sit in your stomach for a long time. So usually for gastroparesis, it's recommended that people avoid fiber. You may have heard of the low FODMAPs, or there's all sorts of modified diets for gastroparesis. But unfortunately, when we try to eliminate fiber, really what comes of that is that we eat a high processed diet. And we also know that fiber is important. It's an important source of prebiotics, which is essentially food for the bugs inside of our gut. And we also know that fiber is important for mitigating constipation. So there's been some studies out of the GI group at Stanford that show that small particle diets, in other words, thinking about things that are easy to mash with a fork, is actually superior to a low fiber diet. So you may want to think about pureeing or mashing vegetables or potatoes, having minced meats. And again, just thinking about anything that's easy to mash with your fork is going to be easier for you to digest. And finally, sitting upright or even taking a brisk walk or exercising to the best of your capabilities. Not that you're going to exercise for 2 hours after meals, but just during that 1 to 2 hour window after meals, staying upright. And if you can, doing a little bit of light exercise, like a walk around the block, will help with digestion. So that was a lot, but to summarize those principles and kind of the big picture diet recommendations: minimize processed foods and sugar, eat a diverse diet. It's been shown that a diverse diet, and specifically a diverse plant diet, leads to a diverse and so-called more robust microbiome. You want to minimize raw vegetables. Eat organic when possible. And really, if you had to focus on any, it's going to be that dirty dozen that you can find on the EWG, or Environmental Working Group website. Try to incorporate fermented foods into your diet, and think about small particle or a diet that's easy to mash with a fork. Okay, so to move from diet to supplements, I generally do not recommend very many GI or microbiome targeted supplements. I am a fan of digestive enzymes. This, again, really can just kind of add more horsepower, so to speak, for you when you're trying to digest your foods. Horse chestnut is a supplement that can be taken on its own, or you can find it in a lot of proprietary mixes that are marketed as GI supplements. One popular one is called Atrantil. But horse chestnut can reduce methane production. So methane, I'll take a step back. So you may have heard of small intestinal bacterial overgrowth, or SIBO. And this is common in Parkinson's patients. It's kind of, there's an association between constipation and SIBO. And we're not really sure, does the SIBO cause the constipation, or does the constipation cause the SIBO? I think it's a little bit of both. But individuals with SIBO tend to have an increase in the bacteria that produce methane, and higher levels of methane causes more gas, more bloating, more constipation. And so again, horse chestnut is an ingredient that's in a lot of GI supplements or can be taken by itself that reduces methane production and thereby decreases gas, bloating, and constipation. Horse chestnut also acts as what's called a saponin, and that binds unwanted chemicals in the GI tract, helping us to eliminate them. And then it also serves as a flavonoid, having antioxidant and anti-inflammatory properties. Glutamine and probiotics. We'll talk about probiotics. So glutamine is something that is commonly used really to try to support that so-called leaky gut or the gut permeability. There is not great evidence to my knowledge really showing a substantial decrease in the gut permeability with the use of glutamine, but again, you may see it out there. I don't think that it hurts. To our knowledge, it's safe. It's safe over the long term. So take it or leave it. And then probiotics. So you're probably aware that there have been kind of a handful of studies looking at probiotics in Parkinson's disease patients specifically. And there have been a few studies, I want to say 3 or 4 since about 2011, that have shown that multi-strain probiotics can increase the number of bowel movements that Parkinson's patients have per week. And then there was one study in 2016 that looked at fermented milk containing, again, multiple strains of probiotics and also prebiotic fiber, again, showing an increase in stool frequency. In practice, my experience, and I was talking to colleagues in GI as well, a lot of our experience is limited by the fact that a lot of patients report actually more bloating and abdominal pain and discomfort when taking a probiotic. And then there is this theoretical risk, though this has never been shown by studies, in part because I don't think anyone has ever studied this over the long term, but there's a theoretical risk that long-term regular supplementation with probiotics may actually alter the microbiome configuration in an unfavorable manner over the long run. So may help increase stool frequency in the short term, but cause some longer term unfavorable changes to the microbiome. So my conclusion and kind of advice on probiotics is that if you fall in the category where you feel okay and you don't have any bloating or abdominal pain or gas from taking probiotics, and you reap the benefit of having more frequent bowel movements, then you probably should just keep doing what you're doing and continue the probiotic. But if you have any of those symptoms, more bloating, more pain, more gas when you take probiotics, or you don't see any change in the frequency of bowel movements, then I would stop taking the probiotic and I wouldn't feel like you're missing out on some, possible benefits. Okay, to address constipation, I wish that I had something new and magical and a quick fix, but I probably don't. We know that hydration is really important. We know that exercise is really important. Again, fiber is very important for helping us to have regular bowel movements. So that's why even though you may have delayed gastric emptying, I do recommend a diet with fiber. Just try to have mashed and pureed fiber. Some patients really like a manual massage. So you can really just put your hands on your belly and kind of go in a clockwise direction. People like to do this at night while they're lying in bed and just kind of gently massage in a clockwise direction around the outer perimeter of your abdomen. And you may find that that helps to initiate a bowel movement in the morning when you wake up. And then some supplements that I love and recommend for constipation. There are many types of magnesium. There's like 7 different forms, but citrate and oxide are the 2 that we like for constipation. This will just help induce bowel movement. So it's really citrate, and you can kind of just honestly play around with the dose that you take. Knowing that it may cause loose bowel movements, then you want to cut back. And of course, stop the magnesium if it causes cramps or any discomfort for you. Other things that you might want to consider, Triphala is a supplement that can be taken for constipation. This is one that it's not a laxative. It really just kind of helps gastric motility. It's a safe one. It's something that can be taken every day forever, as far as we know. And then another to consider is aloe. This is one that you don't want to take every day, and you would want to take just as needed if you're, especially constipated. So with that, I will tell you about the study that we're doing here at Stanford that Tess alluded to. So we are enrolling Parkinson's patients and then also non-Parkinson's patients, ideally someone that lives with a Parkinson's patient. And the idea here is that, as I was showing a couple slides back, there are many things that we know alter someone's microbiome. Living with a pet, whether you drink well water or not, what medications you take, of course, what your diet is like. And so what we're trying to do is kind of create these pairs. So we have a Parkinson's patient and a healthy control that lives with them that presumably has all of those shared environmental factors. And we are collecting blood as well as stool. We're super excited about it because, to our knowledge, this is the only study that has both of those specimens. To date, most people are studying stool, but we're looking at blood, and the couple things that we're looking at are the metabolites. So again, the metabolic byproducts that these bugs make that then go into the bloodstream. But we're also interested in looking at the immune cells and how the activation of immune cells and kind of the inflammatory cascade that immune cells create is different in Parkinson's patients from healthy controls, and which metabolites and bugs might be associated with those changes. So we are going to send a phlebotomist up to you guys to the church on March 11th. And it's just a one-time blood collection. It's about, I want to say it's about 3 to 4 tubes of blood. And then the stool is collected from the comfort of your home whenever works for you. It's 2 little tubes. And then we give you a prepaid shipping label to send it just right back to our lab. And then there's a 20-question survey. And again, that's going to ask you about things like your diet, your medications, your do you have pets, et cetera. And I wish that we could give more as an appreciation, but we're giving everyone who participates a $40 Visa gift card for their time and for their enthusiasm in our research. And so I think what I've talked about for the last 30 minutes really encompasses what we're trying to answer, but some questions that we're hoping to learn from you and from the samples that you provide. So you guys know that there's so much room for improvement when it comes to Parkinson's disease. Right now, we don't have any tests to confirm a diagnosis of Parkinson's disease. It's really the physical exam and up to the clinician's expertise to say that your exam looks consistent with Parkinson's disease. Unfortunately, that means that patients go for a long time where they have probably the early stages of Parkinson's disease and we're not picking them up. And so we're catching people late in the disease course where we don't have as much that we can do. Also, there's a fairly high rate of misdiagnosis between some of the related diseases that you may have heard about, like PSP and MSA, where people, the diagnosis gets mistaken because we don't have some of these more nuanced confirmative tests. So, we're wondering, is there a microbial signature that's predictive of PD? Can we look at someone's microbiome and based off their microbiome predict that they have or will develop Parkinson's disease? Other questions and future goals are symptom management. Can we give more personalized diet advice based off your microbiome? Can we selectively eliminate or maybe add certain bugs or their metabolites in order to address constipation, or even better, to address disease course and treatment? One thing that I'm particularly interested in, and the lab that I work in is particularly interested in, is something called bacteriophages. And these are viruses that infect bacteria. So you can imagine antibiotics are wonderful drugs, but they are very non-specific. So when we take an antibiotic, it pretty much wipes out all of the bugs, wipes out the good bugs and the bad bugs. There is an idea of lytic bacteriophages as targeted elimination of bacteria. So what this means is that using a virus that infects bacteria, and these viruses, what they do is they kind of infect the bacteria, take over the bacterial cell machinery, and then to replicate the virus. And then when they're ready, they lyse or kill the cell and release a lot of the viral replicas. And so you can imagine that this may be a way to selectively eliminate certain species of bad bacteria. So again, these are all just kind of future goals and ideas of where we hope to go with microbiome-based therapeutics and diagnostics for Parkinson's disease. And that is that. So to learn more about our study, I think there is a short form that Mark and Tess will get to you. This is also the contact information for my study coordinator, and you can reach out to her directly. And this is our team. So on the top, this is the team from our research lab. Ami Bhatt is the principal investigator of the lab on the right. And then this is my really wonderful clinic coordinating team. And then on the bottom is the Stanford Movement Disorders Center. So thank you guys so much for your attention.